Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, David Bridey from Oxgene, a WuXi Advanced Therapies company.
Thank you, Leah, for that kind introduction. Today, I'd like to take you back 23 years to 1999. Then, Huda Zogby and her team made the seminal discovery that Rett syndrome, a rare condition that principally affects girls and results in profound intellectual and physical disability, is caused by loss-of-function mutations in the gene for the methyl CpG binding protein MECP2. MECP2 binds methylated DNA, compacts chromatin, and epigenetically silences adjacent promoters. Excitingly, in a murine knockout model of Rett syndrome, expression of MECP2 restores, in part, the cognitive and physical disabilities of mice. Alarmingly, however, even modest overexpression of MECP2 results in a progressive neurological disorder and additional neurological deficits. Moreover, in humans, a simple duplication of MECP2 causes acute neurological and physical disabilities that are quite distinct from those of Rett syndrome. Clearly, in this example, and likely for other conditions, expression of the therapeutic transgene at the correct physiological level is critical to successful gene therapy. Today, I will discuss how Oxgene can help you optimize transgene expression for your systems. Oxgene is part of the WuXi Advanced Therapies group of companies, and we are now pleased to offer promoter discovery for on-target therapeutic gene expression. Together, Oxgene and WuXi's gene therapy pipeline can help clients navigate all aspects of taking therapy from the research bench through viral vector development into GMP manufacture, through clinical trials, and ultimately deliver your gene therapy to the clinic and marketplace. So why do we require novel promoters? Well, I've already given you a real-world example, but in addition, we require promoters that work well in viral vectors. Often, human promoters are too large to fit into the viral vector genome. We require a range of promoter activities to dial in the physiologically relevant amount of transgene expression, because severe overexpression can be toxic. Moreover, control of gene expression may help to relieve the immunogenicity of the gene of interest. Critically, we increasingly require greater tissue specificity to reduce off-target effects. Now, with advances in bioinformatics and genomics technologies, we are in a position to tailor promoter function and activity to your specific cell type and to your viral vector of choice. Oxgene synthetic promoter libraries are generated using bioinformatics-assisted rational design and aided by proprietary in-house algorithms. We're all familiar with the typical promoter architecture, where a core promoter and proximal enhancer comprised of transcription factor binding sites drives gene expression. Now at Oxgene, we have the ability to synthesize a range of core promoters and assemble these with enhancers of combinatorial assembled cis-acting regulatory motifs. We use bioinformatics to identify the component CRMs, using DNA's hypersensitivity sites, which reveals transcriptionally active DNA in open chromatin. This appears as gaps in the sequence reads due to the hypersensitivity of open chromatin to DNase 1. To complement this approach, we also use ATAC-seq data, in which TN5 transposase is used to fragment and tag accessible nucleosome-free DNA. The areas of open chromatin can be identified using NGS and primers against the added tag. Moreover, we combine this information with tissue-specific patterns of messenger RNA expression and the production of single and bidirectional enhancer RNAs from transcriptionally active promoters. From this combined approach, we generate synthetic promoter libraries in the viral vector of choice. Importantly, these libraries are barcoded such that each independent barcode is linked to a unique synthetic enhancer and promoter combination, allowing us to identify individual enhancer-promoter combinations by NGS. The packaged lentiviral promoter library is transduced into the tissue-specific cell type, and fluorescent cell populations are sorted by flow cytometry and single-cell sorting. The process is repeated to enrich for active promoters, and the promoter and relative activity is then determined by RNA-seq analysis and data deconvoluted to identify novel synthetic tissue-specific promoters. Here is an example of an enrichment of a fluorescent cell population from cells transduced with an LV-based library designed to identify promoters specifically active in CD4-positive T cells, such as SUBT1 and JURCAT cells. Through iterative cell sorting and expansion, we can recover populations of cells harboring active promoters. And here you can see an image of SUBT1 cells expressing promoters from the library. Importantly, we observe that many of the recovered synthetic enhancer promoter combinations are expressed poorly in 293 T cells, but are expressed robustly in CD4 positive JURCAT and SUBT1 cells. Moreover, if we take the more active promoters, we find that for the more active promoters, OSP3-23 and OSP54, we observe robust activity in the JURCAT T cell line, but little activity, if any, in a panel of non-lymphocytic cell lines. Thus, Oxgene's synthetic promoters are highly selective for the target tissue of choice. In conclusion, Oxgene synthetic promoters can improve target cell specificity. They can reduce off-target effects and potentially can overcome toxicity of the therapeutic transgene by limiting expression of the transgene to physiological levels. Moreover, we provide opportunities to express therapeutic transgene in response to environmental and developmental cues. Finally, we're able to provide promoters that are uniquely engineered to operate optimally in the client's viral vector and the target cell of choice. So at that point, I'd like to thank you for listening, and if you have additional queries, please contact our business development team at business@oxgene.com. And at this point, I'd like to take questions. Thank you.
Okay, great. Thanks, David. So our first question is, you mentioned that novel synthetic promoters need to be short to be suited to viral vectors. What is the size of the Oxgene T cell specific promoters presented in this webcast?
So the ones presented here, the most active promoters, they were around about 700 base pairs in length, and one of them 1,000 base pairs in length. However, I would say that we can isolate promoters that are much smaller than that. And for viral vectors, for promoters that are destined for viral vectors, our aim is to provide promoters that are below 500 base pairs.
For gene therapy, is it best to go for the most active and robust promoters possible?
No, I think not. I think sometimes there's a drive towards more robust and more active promoters. But sometimes I think it's important to, in terms of the context that the gene is being expressed in, that we aim to have expression at a critical physiological level. And so that's what we hope to achieve. And therefore, with the promoters that we're developing, we'll be able to dial in the amount of activity that we want for that particular gene and that particular tissue.
What viral platforms are you currently using for promoter discovery? Only lentiviral? And can the promoters be easily tested and validated in a different viral system?
Absolutely. So at the moment, I've demonstrated screening in the lentiviral vectors, but we can also do a similar process in adeno-associated virus. And in fact, one of the things that we have been looking at is screening in one virus and then through recombination, move them over into other virus systems and testing there. I think that ultimately the context the context for the promoter is going to be critical for promoter activity. So I think some promoters will work better in LVs, some will work better in AAVs.
Regarding testing and validation of synthetic promoters, will you be able to do a wide panel of cell type testing to both confirm expression in target cell types and show low or no expression in non-target cells?
Yes, absolutely. As I've shown here, we looked at these promoters in lymphoid cells, but also in a panel of non-lymphoid cells. For the client's gene, we will certainly be able to look at similar panels. But I think, that much of the downstream testing is probably better handled under a separate work package and separate SOWs. Thank you.
Okay. It looks like we have time for just one more question. If we didn't get to your question, we will be passing those along to David, and he can answer and follow up with you directly. So please continue to type in any questions you have. But the last question for the webcast is, how long does a typical promoter discovery project take?
So it really depends on the difficulty of the tissue system that you're looking at. For some of the tissues, we already have a lot of bioinformatics available, and so we can push those projects through fairly quickly. I would say that around about 6 to 9 months would be typical for many straightforward tissues. However, for some tissues, it's more difficult because there's a lack of robust data, and therefore we'll have to go out and generate our own bioinformatics data. So those most likely would take a bit longer, and I would expect up to around about a year. Thank you.
Okay, great. Thank you, David.
Thank you, Leah.
And thanks for our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website. And as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.